The overall goal of the following experiment is to screen compounds of interest to determine their cellular cytotoxicity and ability to inhibit wild-type or drug-resistant HIV-1 in a single-round infectivity assay. This is achieved by first exposing the cells to the compounds for uptake, and then incubating the treated cells with luciferase-expressing wild-type or drug-resistant HIV-1. The luciferase activity of the virus can then be measured. Ultimately, the luciferase readout signal can be used to quantify the inhibition of the HIV-1 viral vector replication by the compounds.
This technique has therapeutic implications as it can be used to screen new compounds that exhibit better efficacy against drug-resistant HIV. Such compounds could potentially be used in antiretroviral therapies in HIV-1 patients.
On the day prior to transfection, plate 293 cells on 100 mm dishes at a density of 1,500,000 cells. On the next day, transfect the cells with 16 micrograms of wild-type or mutant HIV and 4 micrograms of VSV using the calcium phosphate method. Approximately 6 hours later, wash the 293 cells twice with PBS and then incubate them with fresh media for 48 hours. 2 days later, harvest the virus-containing supernatants from the 100 mm diameter dishes and clarify the supernatants by low-speed centrifugation for 10 minutes